So let's just wait maybe for a couple of minutes and see if somebody else is going to join us. Okay, so just bear with me for a couple of minutes and see what's going to happen. So how is everything going? You good?
Yeah, I'm good.
Did you email me recently?
Yeah, I
Yeah.
emailed you. I've
What,
got a headache
what happened?
partially.
What happened?
Like sinus.
Oh, sinus headache. I hate those. Yeah.
Yeah.
Yeah. You know what? Yes. Today also I wake up around maybe 4 o'clock in the morning with a very bad headache. You know.
Yeah, my
Yeah.
husband told me maybe it is the weather when the weather change to
Yes, that's true.
yeah, that's why I
Whether
couldn't I
and
couldn't
maybe
study.
allergy. You know, there is a lots of allergen in the air that
Oh,
probably may be responsible for this kind of problem.
yeah, yeah.
Okay, hopefully everything will be fine. So take your time. Don't worry about, I need from you just to feel better
Okay.
so you can do your homework while you are,
Yeah.
in good shape.
Yeah, already I
Yeah,
started today,
yeah,
but
not,
I didn't sit more than 1 hour or 30
yeah, yeah,
minutes
yeah. Don't
like
force yourself. Just, yeah, no,
thank you,
no
sir.
problem. Yeah, it's fine. Yeah, no problem.
Thank you, sir.
Yeah,
Thank
sure.
you. Okay.
Okay, so the meeting today for two things I'm planning to do. One of them is um, talk a little bit about one of the lab which was posted recently about urine culture
Okay.
lab, and then I need to talk about test number 3, which is coming next Tuesday. Okay,
Okay. Okay.
so let's start first with announcement about exam 3. So exam 3 is going to be starting, will be posted Tuesday, and I'm going to post it for a couple of days. So it will be Tuesday and Wednesday of next
Mm,
week.
yeah,
And
sure.
that test is covering 1, 2, 3, 5 chapters, 5 chapters. So it
Mhm.
will be kind of a heavy test. And those 5 chapters are chapter 11, 13, 19, 20, and 21. Okay.
Yeah.
So
Mm-hmm.
chapter 11, which is basically the characterization and classification of bacteria. Chapter 13 is the characterization and classification of viruses.
Mm-hmm.
Chapter 19 and 20 is basically pathogenic, gram-positive, and gram-negative, respectively. And
Mm-hmm.
then 21 is the atypical bacteria. Okay.
Yeah.
Same format like what we used to do before. Multiple choice, true or false, maybe fill in the blank, maybe like a diagram or so, and you need to like some question, for the diagram. And this is mainly the test.
Mm-hmm.
I was hoping that I have more attendance today so we can uh, do some review for those chapters and see if there is any one of those which is kind of hard, some slides which are difficult to understand, so we can go through those together.
Okay.
But I already selected some slides that I need to go through with you as review, okay?
Okay.
That I feel that they are important. For the lab, we have 2 lab assignments this week. One of them is the interpretation of urine culture in real life. That was a very interesting experiment where basically take a sample, urine sample, and try to culture if there is any bacteria in the urine that you need to characterize and count. And that will be an indication if this is like normal flora or that there is a problem or like a disease. Okay. So definitely the bacteria or microflora shouldn't exist in the urinary tract or in the kidney or the ureters. Okay, those are like organ or tissue which should be microorganism free zone. There shouldn't be any microorganism in this like tissue or organs, because the presence of those will be a problem. Okay, and those are some major like bacteria that can cause urinary tract infection. We call it UTI, urinary tract infection. I'm sharing a slide where my presentation. Can you see it?
Yes.
Good. Those are the major kind of microorganisms that can cause urinary tract infection, mainly E. coli, protus, and those are a very good example of gram-negative bacteria. Then you have Staphylococcus, Streptococcus, Lactobacillus. Those are, again, a very good example of gram-positive bacteria. The difference between cocci and bacilli, as you know, cocci are spherical and bacilli are rods. And then you have a fungus, common fungus that also can cause a problem, yeast. Okay, those are some. And how, if you need to grow some of those and test if a sample of urine has those kind of organisms. So in order to do this, there are two different kinds of media that I need from you to know, because those are important, especially for your practical. Okay. The second practical, which is coming in a couple of weeks, definitely this is very important to know. The media, the purpose of the media, the name, if there is any indicator, if there is any specific or special ingredient, you need to know. Two different kinds of media, and those are to isolate gram-negative and gram-positive, but you should be able to differentiate between those. Okay, so when you take a urine sample, you have to use two different kinds of media. One of them, we call it the CNA, or Columbia nalidixic acid blood agar, and from the name, it is a media that has blood. This is why it is red, and it also has a nalidixic acid, the antibiotic nalidixic acid. Okay, this type of media, we call it selective and differential. Selective and differential. So why it is selective? It is selective because it allows only gram-positive bacteria to grow. Gram-negative will not be able to make it. Okay, so we call it selective for this reason. Differential, since it has blood, so it can differentiate between alpha hemolytic, beta hemolytic, and gamma hemolytic gram-positive bacteria. Okay, is why I call it differential. The second type of media called EMB, or eosin-methylene blue agar. It is also selective and differential. It has eosin-methylene blue dye, and it has lactose, the sugar lactose. Okay, it is selective because it prevents gram-positive from growing. It allows only gram-negative, so it selects only for gram-negative. It prevents gram-positive from growing. Eosin-methylene blue is toxic to gram-positive, so they can grow in the presence of those two dyes. Okay, it has lactose, sugar, so it is differential. Differential, so some gram-negative bacteria will be capable of fermenting lactose, and other will not be able to do that. That's why we call it selective. So remember, EMB select for gram-negative, and also differentiate between lactose fermenter from non-lactose fermenter. You look at the media here. If you have a lactose fermenter like E. coli, the bacteria is going to grow in the presence of the media, and due to the presence of eosin-methylene blue, so it will give, if the bacteria is capable of fermenting the sugar, producing acid, the color is going to change to metallic green. So a metallic green color is an indication for gram-negative fermenter, lactose fermenter. If it is like translucent like that, no metallic green, so it is gram-negative, but it is non-lactose fermenter. Okay, so you need to know these. And I think everybody now is familiar with those different kind of hemolysis. Beta hemolytic, this is complete hemolysis. Alpha is partial with this kind of greenish color. Gamma, non-hemolytic bacteria. Okay. So when you do the test, this is a simple lab. Here I need to share with you this slide here, and this is if you take a urine sample. Okay, when you take a urine sample, it is very important to do something called clean catch. A clean catch meaning that you are trying very much to make the area to like decontaminate or remove any kind of natural microflora in the external genitalia first, because you are after those microorganisms inside, okay, that comes straight from the kidney and from the bladder. Okay, so there is a middle, like a clean catch when you allow some urine flow a little bit, that will wash the urethra and will make sure there's nothing there, and then you will catch from the middle where those urine comes mainly from the urinary bladder and or the kidney. Okay, so when you do this kind of experiment and you grow again two different kind of media, CNA and EMB, you have to do two. Okay, and then look at the results after that, how to interpret the data. If you see a picture like that here, that tells you that nothing in CNA, right? It means that there is no gram, there is no gram negative. Okay, and there is no gram negative here because this is select for gram positive. So sorry, it tells you there is no gram positive because we are selecting for gram positive. Okay, so looking at a plate like that, clean, nothing, it means that gram positive is absent. Okay, here you will see only gram negative bacteria because EMB inhibit gram positive, right? So this is an indication that you have urine culture which is gram negative. So this is the first thing. The first thing to tell if you have any bacteria. Is it gram negative or gram positive? No growth in CNA, indication that there is no gram positive at all.
That means it's gram negative.
Yeah, you should look for gram positive here, but as you can see, nothing. So it is mainly gram negative that grows. Okay. So now the second thing you need to, so are those lactose fermenter or non-fermenter? So definitely you have this nice metallic green color. So it means that those are lactose fermenter.
Yeah.
And the second thing, you need to do some count, okay? And because you have a guideline here about the number of colony forming unit per mil, if you have more than 100,000, okay, per mil, it means that, because remember, each colony should come from a single cell, okay? If you have this number, it means that you definitely have an infection with gram negative bacteria. From 10,000 to 100,000, it is kind of doubtful. You need to repeat again and make sure that you have, if you have the same number or you have a different number. Maybe it is at the end of the infection, so bacteria is less now, or maybe it is growing and it will increase later, okay? If the number is less than 10,000, mean that this is nothing, and those are mainly natural microflora in the area, okay? Same here, you grow the urine and you made a urine culture, so you see some bacteria grow in CNA. That means that this is mainly gram positive, okay? Then the second thing you need to look for the hemolysis. Is it alpha, beta, or gamma, okay? Because we know that we can differentiate between different species of Staphylococcus and Streptococcus based on hemolysis, right? And here in this case, nothing grow in AMB, indication that there is no gram negative at all, because you were really expecting to see some colony here, but nothing. So it means that you have mainly collected gram positive from the urine. And now you look for the numbers again and look for those numbers to tell, okay? Again, I can see those two, lots of colony. So this is a urinary tract infection due to gram positive bacteria. It can be Staphylococcus or Streptococcus, okay, or Lactobacillus, for example. This is another scenario here where you can see there is very few colonies on CNA, so this is gram positive. Very few, less than 10,000, so those are natural microflora. If you look at the plate that's selected for gram negative, you can see nothing. So basically, again, you have no gram negative in the sample. This is how you interpret this. So 2 questions for this, very, very straightforward, that you can do easy without any problem. Okay. So do you have any question for me before I move to the lectures now?
No, yeah, it's clear for me.
Okay, perfect. Okay, so let me stop sharing here and go, and I need to share with you. This is this is the virus, chapter 13, and can you see chapter 13?
Yes, yes.
Yeah, this is important, and you know, a very important thing about viruses, things that make viruses different from bacteria, like coronavirus, for example. Many things. They are non-living, you know that. So non-living. So because they can't support their life cycle or their growth without any other living cell. So without any other living cell, they are considered to be like particles made of proteins and genetic materials. Okay, and some phospholipids if they have envelope. They can't do anything unless they are attached to a host, a living cell. Okay, so they are generally non-living. They don't have cytoplasm, the cytoplasmic membrane. They don't have cytosol, no organelles. They can generate energy. Okay, so very, very simple structure, but so dangerous. You know, micro like agent, microbial agents. I can say microorganism, you know, what we can say microbial agents, basically. Okay. What is important? Some slides which are important here about when you look for the virus and viral replication. So virus and viral replication, there is something called lytic and something called lysogenic life cycle. And when you say lytic, this is when the virus attack the host cell, replicate within the cell, and cause lysis of the cell. So the cell will lyse and die. Okay, we call this lytic replication cycle. For any virus to be infectious, there are 5 different stages that the virus should go through to cause a disease and increase in number. First thing, you need to attachment to the host. Okay, here's the first step. And this kind of attachment is mediated by some protein on the virus envelope or the capsid. Okay, if the virus is naked, no membrane, no envelope, and some proteins or glycoproteins or even sugar or fatty acid on the host cell. So this kind of attachment will be followed by entry that will allow the gate to open. And then in no time, either the whole virus will be in or the genetic material will be injected. Then it is very important to start building or making the building blocks. And those building blocks, those are the proteins and the genetic material, nucleic acid. okay?
Yeah.
And then, and this is, again, it happens only in the host. Then that will follow with assembly. So those genetic materials with proteins will assemble to make its virions or viral particles, and then it will be released from the host cell and start a new cycle of infection, okay? And the
Yes.
replication.
Yes.
So this is, we call this lytic cycle. Very, very
Cycle.
obvious in a bacteriophage. And a bacteriophage is a virus that attack bacteria. We call it bacteriophage, okay? And as you can see, attachment,
Virus attack
okay,
bacteria.
attachment, injection of genetic material, synthesis, assembly, and release, okay?
Yeah.
Those are
Yeah.
important. If you look at the, there is a big, huge difference between the growth curve of bacteria,
Yeah.
as we used to see it before. We have 4 different stages, remember?
Yes.
You have
Yes.
lag, which is the adaptation,
First stage. Yeah.
and this is, we have log, which exponential, where the bacteria is growing. Then we have a stationary phase. This is where the number of the dead and living almost the same rate. So cells are dividing and dying at almost the same rate. So this will give you like a straight line like that, kind of. And then finally the cell will stop developing, growing, multiplying, and they will die because it's a death phase. Virus is different. Virus is not following this kind of life cycle.
It's opposite.
Virus, yeah, there is an entry when you have, say, this is the number of phage, virus. Okay, say you have 1 particle, okay? It will enter the cell and then it will be hidden. You'll never be able to detect the virus, okay, because it is hidden in the host. We call this eclipse, the eclipse, because the disappearance, the virus particle, okay? But the virus in this stage is busy making and synthesizing its building blocks and assembling it, okay? And then in no time, at the end of the eclipse, what is going to happen, we call it the burst time. This is where the number will significantly increase. So you will be able to see lots of virions, increasing significantly, okay? And the end of it, we call this is the burst size, okay? So this is the burst time starts, releasing, and the maximum of the number, we call it the burst size, okay? That is going to be, and then the cycle will start again. You have a number here, then you will go down to eclipse again because they are going to infect new cells. It will disappear and then show up again, and so on. This is a very characteristic feature for viruses, okay? Here it tells you about the attachment. There are several scenarios for attachment. One of them is after attachment, there is a direct penetration where the viral particle attach and then leave the coat outside, okay, and get only the nucleic acid in. It is a simple way. So not everything is inside, only the nucleic acid is inside. There is another way where there is a fusion between the viral particle and the membrane of the host cell. Okay, and this is mainly for the enveloped viruses, because enveloped viruses, they have like an envelope made of plasma membrane. So this kind of fusion will lead to release of the envelope will stay out, and the virus particle, the nucleocapsid, and the capsid, and which is the protein coat, and the nucleic acid will get into the cell, and then that will, some proteases, this will be degraded and releasing the genetic material. Or there is also another way of entry, which is endocytosis. And in endocytosis, the whole virion or viral particle will be in through endosome, okay?
Mhm.
So it will bind, and then it will, this is something similar to corona. So they believe
Okay.
that the corona do this kind of thing. Will it bind,
Okay.
and then within an endosome, it will be inside an endosome, and then it will be released. Okay. This is why there is some medication that prevent the release, interfering with the endosome pH and trying very much to prevent the release of the virus. So breaking the cycle at this stage. Okay. So
Okay.
this is very
And
important
ask the
slide.
question,
Yeah.
the difference between the membrane fusion and endocytosis is the caps. This is the code.
Yes, here in membrane fusion, the envelope remain outside. It
On the
fuses,
host, yeah.
with the host plasma membrane,
Yes.
releasing the viral part, okay, which
Mhm.
is the capsid and the nucleic acid. In
Yeah,
the
that's clear.
endocytosis, the whole thing is in.
Inside.
Okay, yes. Okay.
Mhm.
So
Okay.
those are the very important three mechanisms of entry that you need to know.
Okay.
Another thing about the genetic material of the virus, I think this is a very important slide here, that the viruses can be classified based on their genetic materials. Okay, the genetic materials can be DNA or RNA, as you know. If it is DNA, it can be single-stranded DNA or double-stranded DNA. Same deal for RNA. RNA viruses can be single-stranded or
Understand.
double-stranded. Okay,
Yeah.
single-stranded RNA can be plus, and this is plus SS, meaning plus single-stranded RNA, or minus single-stranded RNA. So what is the difference between plus and minus? Any idea?
Mhm. I forget, sir.
I have another attendant who who else was us? Anybody else? Me, you, Membri, and who else?
Mhm.
I think I have another. Hello? Hello? Okay. So anyway, what is the difference between minus and plus?
That's positive strand.
Yeah, what is the
Positive
plus
and
and what does
negative.
minus stands for? So both are single-stranded RNA. But what is the difference between plus and minus?
Positive single-stranded and negative
Yeah, what is the difference? What
single-stranded.
is plus? Is it negatively charged and positively charged? No, it is not positively charged or negatively charged. It means that if the RNA is a messenger RNA that can be translated directly, those are the plus single-stranded. Okay? Meaning that the viral RNA is messenger RNA ready to go. It can be translated directly. Okay? If it is minus strand, then it needs to be converted to plus, okay, to be able to translate. Corona is this type here. So corona inject its genetic material, and its genetic material is plus sense single-stranded RNA. So plus sense single-stranded, minus sense single-stranded. So this is an RNA ready to go for translation. It's like a messenger RNA. In this case, this is a very good example here. Classical example is corona, COVID-19 belong to this. Single-stranded RNA, those are RNA cannot be translated. It needs to be changed to plus to be able to translate. And then you have other viruses, they have double-stranded. So their genetic material like double-stranded DNA, but they have double-stranded RNA. And then finally, there is another group, we call it retroviruses. And retroviruses, they are plus strand RNA, but they prefer to integrate their RNA into the genome of the host. and stay for a little bit before they start like beating the system. Okay, so how those different kind of virus, RNA viruses replicate? If the virus is made of plus RNA, meaning it will release the RNA, which is like a messenger RNA. So that messenger RNA will do two things. One of them is to go straight for translation and making all those kind of proteins. Okay. Oh, and at the same time, with the virus RNA polymerase, it is going to transcribe this messenger RNA and make the complementary. Complementary meaning the opposite. Here it is minus here again, okay? And then another round of transcription for this strand to give you back the plus single stranded, because you need this for packaging, okay? So some of the RNA will go straight for translation and make proteins. Some need to be replicated to make the genetic material. But you cannot make plus from plus. You cannot. So you need to go through intermediate, which is minus, complementary, and then go from minus to plus. This is one group of virus do this. The other group, which is negative, so since they have RNA which cannot be translated, so they need to do transcription and transform it to messenger RNA. And that messenger RNA will give proteins and will go through another round of transcription to make the complementary. So for assembly. Okay? What if the virus made of double-stranded RNA? So you have two strands of RNA. One of them, so it will be unwinding, meaning they are going to be separated. One will act as a messenger RNA, and the other one will be a template to make the opposite. And this one will be transcribed to make the green. So the red will make copies, green copy, and the green will make red copy. And then they are going to reassemble as here. This is important slide, and I'd like, if you have any question, you ask me, please.
I have to study again and again.
Huh?
I have to study again and again.
Yeah, it's okay. I know this is very hard. Sometimes you need to,
Yeah.
you forget it easy, but if there is anything here which is not clear,
It's clear.
ask me, please. This is how we culture viruses. So as you know, you cannot take like a virus and put it in a media like E. coli media, like tryptic soy agar or tryptic soy broth, and ask the virus to replicate. The virus is not going to do this. So what you do basically, you grow the virus and the bacteria. So you have a mixture of bacterial cell and you add the virus and you mix them together, allowing some time for the virus to get access. So it will attach and enter the cell, okay? And you basically what you do, you take this suspension and you make a very nice, you spread it nicely like that on an agar plate, okay? So you start with the, like as if you are growing some bacteria, but the difference is it is a mixture of bacteria and virus together. You spread it nicely like that, and you incubate it for a specific time. So if the virus is capable of infecting the cell, what is going to happen? It will, the bacteria is dividing, right? And at the same time, the virus also is dividing. And when this happened, the cell will die, lyse. And we will see a hollow zone like this. It's like a space, it's like a dot on the agar, which is no bacteria. And this is where you see the viruses. So the viruses are all in these areas here. So we call this viral plaque, Okay, so if you need to get this virus, what you need to do, you resuspend those in some buffer, you put it in this hollow, and then suck it out. You will suck it basically with the buffer and the virus. What happened here, that used to be a colony, but that colony now disappear because the virus eat it. We call it bacteriophage, a virus that eats bacteria. I call it bacteriophage. This is one way to grow them. Okay, and it will be easy to know to do counting. So if you have a suspension of virus and you need to know how many, okay, you can do this kind of experiment, and you, instead of counting the CFU, colony forming unit, you will count something called PFU, plaque forming unit. Plaque forming unit. Okay. Anything in this chapter?
Slide.
You need to know the difference between capsids and capsomeres. And you know that a capsid is basically like the protein coat, and it's made of units. Okay, like you imagine like a room. Okay, the whole room is a capsid, and the wall of the room, like the wall and the ceiling and the floor, those are capsomeres, units. Okay, so you need to know the difference here. Anything in this chapter? Okay, there's another chapter I'd like to share with you, which is 20, about gram-negative bacteria. Okay, so I'm sharing my slide with you. Can you see it?
Yeah.
Okay, one very important thing about gram-negative, as you know, the cell wall structure. And gram-negative, they have two features. One of them is the peptidoglycan layer, which is very thin. And in addition to the inner membrane, they have an outer plasma membrane. This is very unique feature about gram-negative bacteria. What else? That outer membrane is very highly decorated with lipids, fatty acids, and sugar. Okay? And all those basically in the outer membrane structures, those are very virulent agents. Okay? Very important to know if you have like a gram-negative infection and use antibiotics to treat it. You kill the bacteria and you release these outer membrane structures, which can cause lots of problems. Okay, this is what's very wise when you use antibiotic, because you can overwhelm your system with these toxic agents here. So what is unique about gram-negative bacteria? There is something called in the outer membrane. So you have a very nice bilayer phospholipids, as you can see. You have some proteins, integrated proteins, okay? And you have also lipids, and this is fatty acids, okay? We call it lipid A, and lipid A is mainly fatty acids, long chain fatty acids incorporated in the phospholipid bilayer like this. This is like hydrocarbons, long chain fatty acids, and at the top part of it there is sugar, disaccharide diphosphate subunit. This structure we call it lipid A. Remember the name, lipid A. So lipid A made of disaccharide diphosphate, attached to it those long chain fatty acids. Then there is something called the core polysaccharides, and then finally the O antigen. And the O antigen is repeats, around maybe 40 repeats, okay, of these sugar subunits. Those are repeated maybe up to 40 times. Call it, O antigen. So you have lipid A, you have the O antigen, and then you have the core polysaccharide. All those are decorating the outer membrane of the gram-negative bacteria.
Okay.
What is so unique about this? Lipid A is so dangerous. Okay, lipid A is responsible for lots of miserable like symptoms. It can lead to lots of problems. It can trigger fever. So if you have a bacteria gram-negative infection and you have fever, high fever, that is basically due to lipid A. It also causes vasodilation, okay, and redness because it allows the endothelial layer of the blood vessels to be kind of loosen a little bit to allow all those like neutrophils and all those good immune cells to infiltrate out. So lipid A do this kind of thing, vasodilation. It causes inflammation, can cause septic shock. And also there's something called disseminated intravascular coagulation, DIC. And this is clotting, blood clotting,
Blood clotting.
you know, that can also cause by lipid A. Okay, this is a very nice diagram showing how lipid A can induce inflammation, how lipid A can induce fever, how lipid A can induce the disseminated intravascular coagulation. Basically, it activates lots of like immune cells and platelets. So if lipid A activates leukocytes, those leukocytes, well, white blood cells, when they get activated with lipid A, they release several kind of interleukins, okay? And one of them is the tumor necrosis factor. This is, and interleukin 1, those are mainly responsible for fever production. So lipid A induce fever by activating and releasing lots of those like interleukins and tumor necrosis factor, for example. Okay, it can activate blood clotting reactions and activating the platelet factors causing the blood clotting. Okay, blood clotting is very dangerous, as you know, because that will prevent blood flow, especially for organs. So if is the case, that will prevent organs from getting sufficient blood, sufficient oxygen, and sufficient food. So that can lead to organ failure. You know? So lipid A is a nasty structure here, okay? You know,
okay,
the,
so,
yes.
And this, can you go in the previous, yes.
Yes.
Lipid A is in blood, makes blood clotting in 2 ways, that means in your slide.
Yes, exactly.
In 2 ways.
It is basically can go directly and activate those blood clotting agent or activate the platelet activating factors. Yes.
Okay.
Okay. You are not really responsible for like more details about this, but you need to know what is lipid A and what kind of side effects lipid A can do. What is lipid A responsible for in terms of signs and symptoms in disease.
Okay.
This is important to know, and structure for lipid A. So you are not really, you know, you don't need to worry about studying this. No, no, no. That's too much. No. Okay.
Okay.
The gram-negative, we have Enterobacteriaceae, mainly 2 family, and those are most of the human pathogen are within in gram-negative are in Enterobacteriaceae, and we have this in the lab. Remember, we have a very nice identification of unknown from Enterobacteriaceae, and we have Pasteurellacea. Main difference is oxidase reaction. Enterobacteriaceae oxidase negative, Pasteurellacea oxidase positive. Okay, is way to classify those. The other thing here, this is important. I need from you to study this. This is to classify Enterobacteriaceae mainly using all those biochemical data. Okay, you have how many genera? You have lots of genera. All those highlighted in yellow are genera belong to Enterobacteriaceae. And you need to know that the main criteria for classification is basically based on lactose fermentation. Those who can ferment lactose, okay, lactose fermenter, coliform bacteria, and those who cannot ferment, we call non-coliform bacteria, okay? So this is important to know, classification based on lactose fermentation. And motility is another important test for like classification. So one way to differentiate, for example, between Shigella and Salmonella, both are lactose fermenter, they cannot ferment lactose, okay? But the main difference between Shigella and Salmonella, the motility. So Shigella is non-motile, Salmonella is motile, okay? This is important. What is the difference between Klebsiella and the E. coli, for example? So both they can ferment lactose, right? But again, motility. Motility can take non-motile Klebsiella out from the picture, from E. coli. So I need from you to focus on this. This is important slide. Okay, so we need to study this. Yeah, go ahead.
On the last test, you brought this question. I remember the difference of it to differentiate. So, yeah.
Yeah, I don't know why. As I mentioned, because when I put this, there was other chapters.
It confused me. It
Yeah.
confused me.
So now you know. At least if you see the same question, you will be able to solve it. Okay. One thing here, this is a very famous E. coli strain. We call it O157H7. And when you look at this name, what is O, what is H? Any idea?
No, I have no idea. I kind of stuff.
Those are antigen, different type of antigens, okay?
Oh, yeah.
So H antigen is the flagella antigen.
Okay.
And O, this is the O antigen, okay? This is the O antigen.
Yes.
In case of E. coli, we call it O157. So this is mainly, it is made of those sugar, 4 different kind of sugar, monosaccharides, and they are repeated several times. We call it N, okay? And this is very important for like a classification and differentiation of different kind of bacteria. You can do serology based on those antigens, okay? The main problem with E. coli 157, as you know, sometimes it comes to the news if there is meat contaminated with this bacteria. It is a strain of E. coli. You know, most of the E. coli are not harmless. They are fine. You know, they are the major in like normal flora in the intestine of human and other animals, E. coli. But some strains like this one is dangerous because it has some virulence factors that can cause lots of problem. One of them is it can do kidney failure by inducing lots of clots in the kidney. Okay, this is one problem with this kind of E. coli strains. It has it has type 3 secretory system. Okay, and you need to know what is type 3 secretory system. Again, you don't need to worry about the structure, but you need to know the function of type 3. What is type 3? It's one of the virulence factors, or one of the antigenic determinant of some bacteria. Structurally, it is lots of protein subunits together. All those are different protein subunits. They are assembled very nicely in a shape which is like a syringe. So this is the cell wall of the gram-negative bacteria. The inner membrane, the outer membrane, the peptidoglycan. And this is where the type 3 secretory system is sitting It is lots of protein passing this membrane, and some of them in the cytoplasm. And it has a syringe-like structure, like a needle, okay, made of this protein subunit. And then there is another protein subunit, like some holes like that. And this is the host plasma membrane. It's a delivery system. It is like, you know, Amazon Prime and Amazon delivering package. It is the same thing. It delivers effluent and some structure, some virulent factor from the pathogen, the bacteria, to the host. You know, so first of all, they need to attach to the host and then deliver
Then
all those
inside,
virulence factor inside. It's a very dangerous structure. Some pathogenic bacteria, they have this, okay, like Shigella and Salmonella, they have this kind. E. coli, this E. coli here, for some reason, they were capable to get this system, okay? You need to know the difference between Salmonella and Shigella. It is important. I need to focus on the difference between both, because as I mentioned, if you go here, you can see both are non-lactose fermenter, Salmonella and Shigella, but the main difference, the Shigella non-motile, Salmonella is motile, okay? They also they have similarity in the way they infect and attack the human intestine, okay? There is a major difference though. Salmonella, basically, so if for any reason you eat some food which is contaminated with like Salmonella, some animal feces, somebody was dealing with animals and they didn't wash their hand quite well when they were preparing food, or the food was not really cooked very well, so it got contamination with Salmonella. You eat it, it will go all the way through the stomach, all the way to the intestine, where they will find their receptor on the epithelial cell of the intestine, and they will bind, attach. And that attachment is going to follow up with endocytosis. So endocytosis meaning it will get into endosome, okay? The whole bacterial cell will be taken as an endosome. Salmonella, they're unique because they can multiply within the endosome, in the epithelial cell. And when they increase in number, the endosome lice release the bacteria. The main problem with Salmonella is they has the capability to go through the endothelial layer of the blood vessels and penetrate it and go to the bloodstream. And when you have this in the bloodstream, we call this bacteremia. So bacteremia is bacteria in the bloodstream. In the bloodstream, this is a vehicle that will take them to different locations. Okay? Somebody will tell me why they can escape the immune system here. Okay, and I need from you to think about this. Salmonella will go to the bloodstream, but the bloodstream has lots of immune cells. How Salmonella manage to escape from those immune cells and go to different organ and can cause organ failure? And this is what is the main problem with Salmonella. If they were capable of like multiplying and penetrating the endothelial cell layer, then there will be a problem because they will be in the blood. Shigella is kind of different. Shigella also go and bind, okay, and then will be taken, but when the inside, they are not going through endosome. Okay, no endosome. They are going to multiply within the cytosol, okay? And Shigella is non-motile. So in order for them to move from one cell to another, they recruit, they gather actin fiber from the host, okay? And use it as a vehicle to move them from one cell to another, okay? And if for some reason the cell lies and they released to the bloodstream, then phagocytic cells will take care of them and will eliminate them. So they cannot spread like Salmonella. Okay, so kind of less virulent, less dangerous than Salmonella. But the main problem also, they can cause lots of problem with the epithelial cell layer lining of the intestine, causing diarrhea, causing abdominal cramping and fever, because they are gram negative again, so they have lipid A, they can do vasodilation, they can do fever, they can do like blood clotting, all this stuff. Okay, so if you have any question for me for any of those chapters, I will be more than happy to take it. Okay, and now what we can do, just you read those chapters, and if you have any question, I encourage everyone to basically email me if you have a specific question about specific subject, and then we can do another review session before the test. Okay.
Okay, sir.
Okay. Thank you very, very much. Feel better, Memory. Okay.
Thank you.
No problem.
But I have question for the master microbiology class homework.
Say it again. What happened?
For mastering microbiology class, for mastering homework.
Yes. What about
on Pearson for chapter 24 and 25. Yes. No, chapter 2. What you gave us the last chapter. 19 and 20 something. I did it, but I have question on number 24C.
Yeah, can you email me that one if you don't mind?
Okay.
Yeah, email it to me, and then if I can, I will maybe I will record like a very short Zoom and email it to you so you can see my response.
Thank you, sir.
Okay.
Thank you, sir.
No problem. Thank you very much, guys. And you have a good night and feel better.
Thank you, you too. Thank you.
Thank you. Bye bye. Bye bye.